Happy Friday, everyone. So, last time in getting ready for metabolism, I introduced the topic of Gibbs free energy. And Gibbs free energy is a very important consideration for metabolic processes, the reason being that, as I said last time, energy is a consideration in every reaction that's run. And the Gibbs free energy change in particular is important because it tells us the direction of a reaction. Now, one of the things that we will see is important in metabolic processes is that many of them, not all of them, but many of them go forwards and backwards. And the direction that a process or a pathway goes depends almost solely on the concentration of products and reactants. And so that's what we see in this top equation, delta G being equal to the delta G0 prime plus RT times natural log of products over reactants. As I mentioned last time, the only real variable there is the products and the reactant concentration. So as those products and reactants change in concentration, the direction of a reaction can actually change as well. When we look and talk about glycolysis, which will be our first metabolic pathway that I'll talk about in just a little bit actually, we see that glycolysis in the forward direction is the breakdown of glucose to produce pyruvate and some other molecules for metabolic energy. But what we will discover immediately after glycolysis is that there's a reversal of that process called gluconeogenesis in which glucose is made. And in that process, most of the reactions of glycolysis, not all of them, but most of them, are the reversal of the reactions that happen in glycolysis. So it tells us that cells have the ability to change concentrations of things, and in doing so have the ability to determine directions of reactions. And as we will see, the only real control that cells have over their destiny is the control of the concentration of products and reactants. So that's a very important consideration. As I mentioned last time, as the concentration of products increases, the value of this ratio increases, which makes this term more positive. And the more positive this term is, the more positive the delta G will be, which will tend to favor the reversal of the reaction. On the other hand, if the reactant's concentration increases and the product's concentration does not increase, then this ratio becomes more negative. And more negative here makes delta G itself more negative, and more negative delta G means more likely to go in the forward direction. So the direction is really, really important. I also want to point out the delta G0 prime term here. And I do that because I find that students have a tremendous amount of confusion between what delta G is and what delta G0 prime is and what they tell them. That is what each entity tells you. The delta G, as I said, is the only thing that tells you the direction of a reaction. Delta G0 prime does not tell you the direction of a reaction. Okay? Delta G0 prime is a constant for a reaction. It's a constant for a reaction. So if I'm talking about glucose going to glucose 6-phosphate, then I have a delta G0 prime value associated with that, and it will always be associated with that, just like we saw that a pKa value was always associated with the same weak acid. Okay? So it's a constant. Delta G0 prime does not determine the direction of a reaction. Delta G0 prime influences it because it's one of the two terms that contributes to the delta G, but we can't consider the delta G unless we consider both of these terms. So if I try to tell you on an exam, for example, the delta G0 prime for a reaction is negative, I don't want you sitting here and telling me that reaction is going forwards because the delta G0 prime is not the only consideration. Okay, so you have to consider both of those values in determining the overall reaction direction. Okay. Well, as I mentioned last time, the delta G0 prime is equal to the delta G only when we're at standard conditions. And when we're at standard conditions, products equals reactants equals 1 molar. So when that happens, 1 over 1 is equal to 1. The natural log of 1 is, of course, 0, which means that this term disappears when we're at standard conditions. And, of course, at standard conditions, as I said, delta G0 prime is then equal to the delta G. That's the only time that happens. Okay? So we don't have other conditions under which that overall process happens. Allergy season is here, isn't it? Okay. Well, I'd like to show this diagram because I think it shows a bit about the flow of metabolic energy, ultimately starting with the process of fusion in the sun that gives rise to the radiation that hits our Earth. That radiation is captured in the form of chemical energy by the process of photosynthesis, in which carbon dioxide and water are assimilated to make glucose and oxygen. The oxygen that we have in our atmosphere is largely a product of photosynthesis that's been going on for billions of years on the face of the Earth by plants. Glucose and oxygen are important because, of course, oxygen is what we breathe and we use as an electron acceptor, and glucose is necessary for metabolic energy. So these are used through a process of oxidation to make ATP out of ADP. So ADP has less energy than ATP does, and so if we want to make a high energy molecule, we have to use some energy to do that process, to put that in there. Just like if we have water down at the base of a hill and we want to get it to the top of a hill, it will not flow up by itself. We have to put energy into raising that water to the top of the hill, and that's what's happening right here. We can also use the energy from ATP to make things inside of cells. ATP is necessary for making most of the molecules in our cell. We've seen one exception to that, and that exception being proteins, where GTP energy is used, but it's to simplify this figure that we have ATP here. The energy of ATP is exactly the same in terms of the quantity of energy as we find in GTP. So, starting with the sun, we ultimately realize that energy in the form of complex biomolecules that we're able to synthesize in our bodies. Also want to introduce a couple of terms here, catabolism and anabolism. So catabolism, or catabolic processes, are processes that involve breakdown of bigger molecules into smaller molecules. That process of breakdown of bigger molecules into smaller molecules usually involves oxidation. Okay? Doesn't always. The breakdown of proteins into amino acids does not result in oxidation, but the other processes ultimately do result in oxidation. And catabolic processes are, in general, as a consequence, energy releasing. We capture that energy in the form of ATP. Anabolic processes, on the other hand, are synthesizing larger molecules from smaller molecules. So anabolic processes are the opposite of catabolic processes. Consequently, they tend to be reducing in nature, which means they require electrons instead of producing electrons, and they are also energy requiring. So when we go and we eat a cheeseburger, our body takes and breaks down all of the protein that's in there in our digestive system using proteases, some of which you have seen. And we take the amino acids that result from that breakdown of the proteases and use those amino acids to make our own proteins. And to make our own proteins from those amino acids, of course, requires energy. So that's why having this ATP from oxidation is very, very important. At any given time, we have catabolism and anabolism going on in our body. Okay? Our body is continually doing both processes, and managing that takes a considerable amount of effort, as we will see, within a cell. Okay? Carbon is, of course, the central molecule in organic molecules, and an essential, not molecule, central atom in organic molecules, and essential for a life as we know it on the face of the Earth. It's important to recognize that carbon has several oxidative states, this state on the left here being the most reduced, and each step over in this direction becoming more and more oxidized until we get to carbon dioxide. Carbon dioxide is, of course, the ultimate product that we make of the oxidation of carbon-based compounds in our body, and we exhale carbon dioxide because it actually is a poison to us. The process of oxidation is one we've talked about. I've talked about briefly in class, but I want to make sure everybody's on the same page, so I'll repeat a little bit of this again. If I have a molecule that's an alcohol, in this case ethanol, and today's a great day for happy hour, by the way, and I oxidize that ethanol, I make my hangover molecule, acetaldehyde, over here. Okay? And to do that, that involves an oxidation step because I'm converting an alcohol into an aldehyde. And you can convince yourself of that if you do what I did here, where you count the electrons, and you see that this guy had 12 electrons, and this guy has 10 electrons over here. That meant that 2 electrons had to disappear. And they didn't disappear. Of course, I'm showing them on the equation. But biologically, those electrons get picked up by electron carriers. I've talked about that in class previously. And we'll see something about that. These reactions are reversible. Okay? So an oxidation, right, can be reversed in the form of a reduction if electrons and protons are available for that reduction. We have several electron carriers that our body uses to accept electrons arising from oxidative processes. And one of the most, in fact, the most common of these are the nicotinamide-based electron carriers. Here is a molecule of NAD, and NAD is lacking a couple of electrons. If we reduce that with 2 electrons, we go over here and we make the reduced form, and you can see that the ring structure has actually changed because this guy has gained 2 electrons. Now, the biological oxidations generally involve 2 electron transfers. And that's good because NAD accepts electrons 2 at a time. When NAD accepts 2 electrons and 1 proton, it becomes NADH. Now, NAD starts out with a plus sign. NADH has a charge of 0. So how does that work out? Well, that works out as follows. If I take something that has a +1 charge and I add 2 electrons to it, what will I have in terms of charge? I take +1 and I subtract 2. I would have a -1, right? But I put a proton, 1 proton on there, to that -1, I end up with an NADH that has a charge of 0. Okay? So that's why we actually write this thing as NADH plus H+, meaning the second hydrogen doesn't go on to the NAD to make NADH2. We don't have something like NADH2. But we convert NAD from a +1 charge into a 0 charge through what I just described. Another electron carrier in cells is that of FAD, and the FAD is attached to something else down here. It's just designated by an R. The relevant portions of the reduction are shown here, in which we are going from this guy on the left over here to this guy on the right. Okay, so that is, yes, the rearrangement here. So count the electrons and you'll see. So 2, 4. That actually is backwards, isn't it? What do we have? We have, no, I'm sorry, I got to count this down here. Okay, so there's the addition of the protons on the electrons up here. You're not going to have to draw these structures, so don't worry about that. The reduction of FAD goes to make FADH2. In this case, 2 electrons and 2 protons go on. We start with a molecule FAD that has a charge of 0, and we end up with a molecule FADH2 that also has a charge of 0. Now, these reactions are also reversible. We can go from one to the other. This simply shows the overall structure. I didn't draw you in the last figure, but there's that top portion up there, and here's a molecule of AMP. And if we actually included that right there, we would have a molecule of ADP. So ADP, as we will see, is a very important molecule in a lot of various biological molecules. Well, ATP, of course, is the molecule we refer to as the gasoline of the cell. It is the energy source. It's the form in which energy is stored in the cell. And it's an immediate source of energy. There are other sources of energy storage, glucose being one of them. Glucose is a storage form of energy, but the energy in glucose has to be oxidized through the process of glycolysis before the energy can be realized, whereas the energy here in this ATP is immediate. If we need to go run a race or jump or do something like that, then what we have is a need for ATP because ATP provides the necessary immediate energy for muscular contraction. Why is ATP a high energy molecule? Well, the answer to that question is actually rooted out here. Okay, if we look at the 3 phosphates that are out there, you probably learned all along that, well, the 3 phosphates have more energy than the 2 phosphates, but probably nobody told you why the 3 phosphates had more energy than the 2 phosphates, so I'm going to tell you here. Phosphates, of course, are negatively charged, and negative charges repel each other. You ever try to put together two north poles of a magnet? What do? They don't like each other, right? Well, you try to keep together these negative charges, they don't like each other either, okay? So when you break a bond, for example, breaking a bond right here, this negatively charged phosphate goes, I'm out of here, right? You actually can get motion out of that. You actually release heat as a consequence of breaking of that bond. Okay? And that simply shows ATP, ADP, and AMP. It takes water, hydrolysis reaction, to break off a phosphate. That's the water right there. And we can see where the water, actually we can't see where it's added there, but we can see where it actually targets the breakage right there. Water coming in right there. Okay, so these are some molecules that we're going to be talking about a little bit in the days ahead. And you can see these guys have some incredible energy. Okay, the more negative a number is here, so these are DeltaGzero prime values, right? So the more negative a DeltaGzero prime value is, the more influence it's going to have on that delta G equation. And it means that if I start out with equal concentrations of products and reactants, for a reaction that has this negative of a DeltaGzero prime, it means that that reaction is going to go very far in the forward direction to reach equilibrium. That's really what DeltaGzero prime means. So therefore, if I have that case, this guy is going to go very far forward, the hydrolysis of this guy, to release a lot of that energy. Now, if you look at these guys, you'll see, for example, that PEP has about twice as much energy as there is stored in ATP. You say, oh, well, PEP would be a great energy storage form, and I suppose it might if things had evolved that way, but the problem with that, the bigger question that I have is, well, it turns out that how do we make PEP? Okay? We could actually use ATP energy to make some of these molecules, but how do we make something that has more energy than ATP with something that has less? How do you suppose we would do that? So John says you could use multiple molecules of ATP, and that's not a bad answer. But there's actually a simpler answer, and the answer is right here in this equation. How would I do that? How would I make a reaction go forwards, okay, if I've got essentially a positive delta G that I'm starting with? What's that? So by altering the concentration of products and reactants, okay, I can actually make that happen. Now, we don't make PEP that way, so that's kind of a bad example, but there's one of the molecules on there called creatine phosphate. Creatine phosphate is a material that's in our muscles. And in our muscles, okay, creatine phosphate takes more energy to make than ATP releases. Well, how do we make creatine phosphate? Okay, let me see. I think I've got, I don't have that on here. Okay. How do I make creatine phosphate? Well, to make creatine phosphate, I've got a simple reaction. I've got creatine plus ATP makes creatine phosphate plus ADP. The phosphate goes from the ATP onto the creatine. That reaction goes forward in our cells, even though there's less energy in ATP than there is in creatine phosphate. Well, the way I make that happen is by increasing the amount of reactants, in this case creatine plus ATP, and decreasing the concentration of products, which is creatine phosphate plus ADP. Now let's think about this. Creatine phosphate's really important because our muscle cells use it as a backup source of ATP energy, right? If I go off and I decide to go do a 100-yard sprint, which for me is not very easy to do, but if I decide to go do a 100-yard sprint, I can burn up most of my ATP in the first 20 to 30 yards. If I can only go 20 or 30 yards on what I had, that grizzly bear that I worry about catching me is going to catch me, right? So I have a backup, and the backup is creatine phosphate. Creatine phosphate is a high-energy compound. And from the reaction I just told you, Think about it. I want you to think about this reaction. Creatine plus ATP goes to creatine phosphate plus ADP. If I run that reaction backwards, what do I make? I make ATP. Creatine phosphate plus ADP gives ATP plus creatine. Creatine phosphate is a great way to store energy so that when I start running out of ATP, which direction is the reaction going to go? It's going to go backwards, right? And when it goes backwards, what do I start making? ATP. So how do I make it go forwards? Well, I make it go forwards by sitting around, eating beer, drinking beer, eating pizza. I got the two mixed up. Eating beer, eating pizza, drinking beer, right? And watching the tube. I'm not burning energy. What happens to my ATP levels? They go up, right? ATP levels go up. What happens to that creatine plus ATP equation? It starts making creatine phosphate because I have more reactants. And when I take off on that race, the ATP levels drop, and it goes backwards, so I can make ATP. It's a very cool setup. Yes, ma'am? Is there a limit to how much creatine phosphate you can make? Yes. It would depend on how much creatine you have. It would depend on how much ATP you have, because those are products and reactants consideration in this equation. So yes, there absolutely is. Yes, John? Or what toxic for the cell? So the creatine, as long as you're not messing with things, I'd say no. The problem is people decide that they want all this athletic performance. And somehow people have equated athletic performance with healthy living. And it's a big mistake. Okay? Athleticism, yes. Athletic performance, no. All right? There's a difference. So people will go out and say, oh, I'm going to take all this creatine. Right? I take all this creatine. All right? Let's imagine this brainchild that just decided I'm going to go out and I'm going to go run a race. And I'm going to take a whole bunch of creatine because it's going to help me to have all kinds of energy because now I can make more creatine phosphate, right? Well, let's imagine I did it just before the race. Here's my equation. Creatine plus ATP goes to creatine phosphate plus ADP, right? How readily is that reaction going to reverse if I have an excess of creatine? I'm going to go about 20 yards and I'm going to go boop, right? I'm not going to have the backup source that I want. So in answer to your question, all right, in theory, they're not toxic, but they're not something I would mess with, right? Athletic people doing athletic performance insist on messing with that. And I think it's not a good idea. So I strongly encourage that. I strongly discourage that. I should be clear. Okay. ATP is, of course, the central figure in all these processes. Catabolic processes are making ATP. Anabolic processes are using ATP. Okay? So we have to be able to make it, and we have to be able to use it to function as a living organism. This is a system we're going to be investigating over the next few lectures. The top part basically being the process of glycolysis, which is right there, and this process of the citric acid cycle, which uses the output of glycolysis to do a lot of oxidation, producing carbon dioxide and a variety of molecules of NADH. We'll see how the cell uses that overall to accomplish what it does. These processes are reversible to some extent, and so catabolism and anabolism require balance, and we'll see how the cell uses a complex set of regulation systems to actually achieve that balance. Okay. So that's what I want to say about energy. I want to dive now into talking about the process of glycolysis. So glycolysis is again, the name tells us what it does. Glyco meaning sugar, lysis meaning breakdown. So glycolysis is the process of breaking down a sugar, specifically the sugar glucose. We'll see later how other sugars can be converted into intermediates in the glycolysis pathway so that they too can be oxidized. Now I'm not going to start up there with pyruvate fates. I'm going to come back to that later. I've got that really in the wrong place. This figure shows the overall process of glycolysis. Okay? First place, I'm not going to ask you to memorize any structures. All right? So no structures for memorizing there. I do think you should know the names of the enzymes. And with one exception, every enzyme tells you what it does. Its name tells you what it does. One exception. Okay? There are 10 steps in the process of glycolysis. We start with a molecule that has 6 carbons. And at the end, we have 2 molecules that each have 3 carbons. Okay? We produce a little bit of ATP energy. We see it down here and we see it down here. But glycolysis is unusual in that it requires energy to start the process. Okay? So we have to input energy in order to get the glycolysis process started. We can see the 6-carbon molecule gets split into 2 molecules of 3 carbons each, and that's shown down here. And then from this point downwards, everything's identical for each of those. So we essentially have 2 of these, we have 2 of these, 2 of these, etc. The overall process of glycolysis requires input of 2 ATPs, and on the other side it gives 4 ATPs. So there's a net production of 2 molecules of ATP. There's also net production of 2 molecules of NADH. You can see them right here and right here. That means that there's an oxidation that occurs in the pathway. In fact, there's 2 oxidations, but since they're 2 identical oxidations, we talk about it as if it's one, but they're 2 identical oxidations that are happening there. And most importantly, at the very end, we also produce 2 molecules of pyruvate, because these molecules of pyruvate are then used in the pathway of citric acid cycle to produce a lot more energy through oxidation. So a lot of glycolysis, the function of glycolysis is simply to provide intermediates necessary for further oxidation elsewhere. Okay. Now, I'm going to go through and tell you the reactions. You should know, you should be able to name the reactions and to name the enzymes. I'm not going to make you memorize structures. Okay? Name the molecules and name the enzymes. The first phase of glycolysis is called the energy investment phase. And that's where the ATP energy is necessary to get the process started. Okay? It's not overly complicated. Let's look and see what happens. We've got a molecule of glucose. Okay? We phosphorylate it. We rearrange it. We phosphorylate that, and we split it in 2. That's what's happening in the first phase of glycolysis. Phosphorylation, rearrangement, and splitting up. Phosphorylation, rearrangement, additional phosphorylation, and then splitting it in 2. That's what glycolysis, how glycolysis starts. Wherever you hear the word phosphorylation, that means that a kinase is involved. And phosphorylation by a kinase requires energy of ATP. So we start with glucose. We put a phosphate onto glucose. Where does it go? It goes on to position number 6, and we have glucose 6-phosphate. We isomerize that to make a molecule of fructose 6-phosphate. So we've gone from a glucose 6-phosphate to a fructose 6-phosphate. We phosphorylate that fructose 6-phosphate using another kinase. This one's called phosphofructokinase, or as you're able to call it PFK. PFK is a kinase, and we'll see it's a very important enzyme in the regulation of the process of glycolysis. PFK. The product of that gives a molecule of fructose 1,6-bisphosphate, and I prefer to call it F16BP, not FBP, but F16BP. You can see the phosphate there and the phosphate there. John? First kinase, I'm sorry. I may not have even said it. It's hexokinase. Okay, so let's look at what the names tell us. Hexo means 6. 6-carbon kinase. It's putting a phosphate onto a 6-carbon kinase, into a 6-carbon molecule. The product is glucose 6-phosphate. The second enzyme I didn't mention either is glucose phosphate isomerase. Glucose phosphate, isomerize it to make another molecule, right? This enzyme is phosphofructokinase. Again, phospho, it's a phosphofructo molecule because there's the fructose, there's the phosphate on it. Phosphofructokinase, or PFK, uses energy from ATP to make ADP, and we make F16BP. Now, this guy, this F16BP, is split in half in the next reaction. Split in half. At this point, we get 2 molecules of 3 carbons each. Each one of them has a phosphate. The enzyme that catalyzes this reaction is they're all important enzymes, but this one's an interesting enzyme. It's called Aldolase. It's the only enzyme whose name doesn't tell us what it does. The only enzyme whose name doesn't tell us what it does. Yes, ma'am? Aldolase. A-L-D-O-L-A-S-E. Aldolase. Yes, Maria? So just get into that. So the 2 molecules that are produced by the action of aldolase are, I'm just going to give you the abbreviation because that's probably what you're going to call it, is DHAP, dihydroxyacetone phosphate. P. P is in phosphate. The other molecule is known as glyceraldehyde 3-phosphate, okay, or what I like to call G3P. Yes, sir? The 2 that come out are not the same. One gets changed into the other in the next reaction. So DHAP and G3P. G3P, glyceraldehyde 3-phosphate. Yes, sir. So the F16BP is the substrate, right? So the F16BP is the molecule that gets split into the 2 3-carbon pieces. The enzyme that does that is known as aldolase. So we've had 4 enzymes so far. Hexokinase, and I'll mention glucokinase later. You don't need to worry about that. But hexokinase, glucose phosphate isomerase, PFK, and aldolase. So at this point, we have completed the energy investment phase of the pathway of glycolysis. Completed the energy investment phase of the pathway. Now, one of the things I'm going to tell you, and we'll talk about later, is the fact that this last reaction that I showed you is energetically a big hump for the cell to get over. It's a big hump for the cell to get over. The delta G 0 prime for this reaction is about positive 20 kilojoules per mole. It means if we start with equal concentrations, this reaction is going to be favored strongly backwards, and we want it to go forwards. So this is going to take some manipulation of products and reactants by the cell in order to make this reaction go forwards. The cell does it with a very cool trick that I'll show you later. Questions on that before I go forward? Is that a hand? Oh, you're just stretching? Okay. All right. There's the hexokinase reaction. Now you can actually see these guys a little better. You're not responsible for the structures, but I just show you this to show you what's happening. There's carbon 6, and that's where the phosphate's going on. Okay? This reaction, in the absence of ATP, can actually occur. But in the absence of ATP, when you're putting a phosphate onto here, it's energetically very unfavorable. Okay? It's very energetically unfavorable, which means we need energy to make this thing go forward. Where does the energy come from? Well, when we use ATP, we hydrolyze that phosphate, and it gets transferred directly onto the glucose. And this, when ATP is present, okay, when ATP is present and this reaction goes, it is energetically quite favorable. Okay? This is the enzyme I think I've mentioned earlier in the class that has the unusual property, I shouldn't say unusual, but it's one of the flexible enzymes. And its flexibility is changed by the binding of substrate. I like to think of it as two hands. Top hand binds the glucose, bottom hand binds the ATP. When the process starts, the enzyme literally looks like this. One substrate binding site, other substrate binding site. When it binds the two substrate, the jaws close. That puts the phosphate into close proximity with that carbon, and the phosphate just jumps. That's all that happens. And when the phosphate has jumped to make the G6P, the jaws open, and the products are released. Okay? So it's a very cool process. We can see very easily how the flexibility of the enzyme facilitates this overall reaction. Okay. It's G6P. G3P is glyceraldehyde 3-phosphate. So G6P is glucose 6-phosphate. Okay? Okay, there's glucose 6-phosphate, and that's G6P. You can also call fructose 6-phosphate F6P if you want. Okay? F6P. So that's G6P and F6P. This is a simple rearrangement. We're going from glucose 6-phosphate to fructose 6-phosphate. Nothing else has changed. There's glucose phosphate isomerase. Here's a reaction that's very much like the hexokinase reaction. We try to do it in the absence of ATP energy. It's not going to be favorable. If you want to put the phosphate on, it's just not very favorable, but we put ATP energy in, and it becomes very favorable. Both of these reactions are examples of something we call energy coupling. Energy coupling. Energy coupling occurs when an energetically unfavorable reaction is made favorable by combining it with an energetically favorable hydrolysis. So energetically unfavorable coupled with an energetically favorable one makes the overall process favorable. That's energy coupling. Yes, question? The magnesium. Oh, so magnesium is just an ion present in the cell. Almost any time you have ATP involved, magnesium is needed because magnesium chelates into the phosphate. The positive charge chelates into the phosphates of the ATP. Okay, so at this point we've made fructose 1,6-bisphosphate. We're getting ready for the big split. Okay. Let me come back to that. Okay, the big split. Fructose 1,6-bisphosphate splits into dihydroxyacetone phosphate. That's the DHAP. Glyceraldehyde 3-phosphate. That's the G3P. Okay. This reaction, as I said, is very energetically unfavorable. The DeltaGzero prime, very positive. So it means if I start with equal concentrations of everybody at equilibrium, this guy is going to go backwards. Well, it tells us that in the cell, if we want it to go forwards, we can't start with equal concentrations of everything because the reaction is going to go the direction we don't want it to. Like I said, the cell uses a very cool trick to overcome this.
So the cell uses a cool trick to overcome this. Now, does the cell also use that similar idea in combination with energy coupling?
Okay, so the cell is not using anything to do with energy coupling here. No. So, but let me save telling you what the cool trick is, because I want to go through the whole. In order for you to understand the trick, you've got to see the whole pathway. Okay. Was there a hand? Okay. Okay. Now, in the next reaction, the cell decides to do students a favor and convert DHAP into glyceraldehyde 3-phosphate, which means that from that point forward, everything is two copies of the same thing. Otherwise, you'd have to learn two separate pathways. And the cell, for some reason, decided to be nice and not make that happen. Okay. So the enzyme here is an interesting enzyme. It's called Triose phosphate isomerase. You can call it TPI if you wish. And TPI is an example of what we call a perfect enzyme. Did I talk about perfect enzymes when I talked about enzymes previously? I don't think I did. What's a perfect enzyme? A perfect enzyme is an enzyme that takes very little substrate and has an enormous energetically high reaction rate. Okay? That's what a perfect enzyme is. This enzyme is an example of a perfect one. And you might say, well, why do we have a perfect enzyme here? Why have that going on? Okay. Well, the reason we have a perfect enzyme here is we want this reaction to go through as quickly as possible, because it turns out that this process uses a molecule that as an intermediate between the two, going from here to here. And the intermediate turns out to be poisonous to the cell. If the intermediate gets released, the cell is in trouble. So to reduce the likelihood that the intermediate will get released by the enzyme, the cell does the reaction. The enzyme does the reaction as fast as it possibly can. Okay. Very cool evolutionary process that made that. The molecule, I think it's called methylglyoxal. Yeah. Yes, sir.
Yeah. Could you go through the perfect enzyme one more time, please?
Sure. A perfect enzyme is an enzyme that has extraordinarily high kcat and extraordinarily low Km, meaning it doesn't take very much to get to really high rates of action. Okay? Okay. Well, as I say, this way now we've got two molecules of glyceraldehyde 3-phosphate. So from this point forwards, everything will be identical. And notice here it says first phase, and the diagram I showed didn't show that TPI in the first part of the process. You can put this reaction in either the first phase or the second phase of glycolysis. It doesn't really matter. Yes, ma'am.
Was that reaction a part of the energy investment?
That's what I'm saying. Yeah. So it's actually, I just noticed here, but this says first phase, and in fact, you could put it in second phase. The diagram didn't show this reaction. So it doesn't really matter because it doesn't involve energy investment and it doesn't involve production of energy. So it could go in either one. Okay. In fact, some people actually put these two reactions into a third category, which I don't do to keep it simple. Okay. Okay, well, we're ready for the second phase, and it's in the second phase that the oxidation occurs. It's in the second phase that ATP is produced, and we'll quickly go through that. The first reaction is a very interesting reaction. First reaction involves an oxidation. NAD is going to NADH. You can see that an aldehyde is going to an ester. That means it had to become an acid in the process. So that's an oxidation reaction. And we see the addition of a phosphate onto that acid. Now, I told you when I tried to put the phosphate onto glucose, it didn't go very well. It needed ATP energy, right? I told you when I tried to put the phosphate onto fructose 6-phosphate to make fructose 1,6-bisphosphate, it didn't go very well. You needed ATP. My question to you here is, why am I able to add a phosphate here? What's different? There is something different. There is a point of extra credit. So there's an oxidation going on here, right? I just told you the answer. Oxidation produces energy. This oxidation reaction produces enough energy to use to put that phosphate onto here. Okay? So this is an energetic reaction, energetically favorable reaction in the forward direction. And it creates a molecule that actually has high energy. If you looked at that table I showed you earlier where I had those high energy molecules, this is a good example of one right here. We'll see that happen in the next process. So this reaction is giving us a high energy intermediate. It's also giving us NADH. And remember that since we have two of these and two of these, we also have two of these. So this is where we get the two NADHs from the process of glycolysis. This is the only oxidation that occurs in glycolysis, the only oxidation that occurs in glycolysis. Maria?
Does the enzyme make what makes oxidation, or what causes
This is the enzyme that catalyzes the reaction that involves that oxidation. That's right. And by the way, that's a long mouthful of a name. You can call it G3PDH. G3PDH. G3PDH catalyzes the only oxidation in the pathway of glycolysis. How am I doing? Okay. In the next step, we see that our first production of ATP. Okay? This production of ATP happens by the transfer of this phosphate onto ADP to make ATP. This is a relatively unusual reaction. And it's relatively unusual because we don't usually make ATP by transfer of a phosphate directly onto it by another molecule. Okay? This is an example of what we call a substrate-level phosphorylation. A substrate-level phosphorylation. And a substrate-level phosphorylation involves just what I said. Transfer of a phosphate from a molecule onto ADP to make ATP. Most of the ATP that's made in cells is not made in this way. Most of the ATP that's made in your cells is made by a process called oxidative phosphorylation. And I'll repeat that because that's a very important name. Oxidative phosphorylation. And there's a third type of phosphorylation that cells use to make ATP. It's not used by us, but it's used by plants. It's called photophosphorylation. As its name would suggest, it uses light energy to catalyze the formation of ATP. We'll talk about it with photosynthesis. So 3 ways that cells have of making ATP. Substrate-level phosphorylation, oxidative phosphorylation, and photophosphorylation. This is an example of substrate-level phosphorylation. The enzyme is phosphoglycerate kinase. Notice it's a phosphoglycerate. Glycer referring to glycerol. This is related to glycerol. Okay? Kinase, putting a phosphate onto something. What's it putting a phosphate onto? It's putting a phosphate onto ADP to make ATP. It's an unusual kinase in that respect. Yes? For the reactant? Oh, 1,3-BPG. That's going to sound like something we've talked about, right? And guess what? 2,3-BPG doesn't enter the equation yet. But it sounds like it, right? The product is called 3-PG. 3-phosphoglycerate is 3-PG. Okay, so here's what we're going to do. I wasn't going to do an extra credit today, but I've decided you guys have been very patient and quiet. Quiet not being the good part. I like when you interact more. So I will allow you an opportunity to do extra credit. I want to do something creative here. What should we do creative to get extra credit? Well, we'll be here for too long, I'm afraid, if we do that. Okay. Do the following. Let me think. Do the following. Put your name and ID number on a piece of paper. And if you want extra credit, you must email me a question to consider for the next exam.
So what's the point of this?
So I know you're here.
Oh.
Right?
But people who work here want to email you unless
They won't have a piece of paper from them. Right? So only those who are here will have that piece of paper turned in. So I'm getting a piece of paper from you with your name on it, ID number, and a question for the next exam. I will pick at least 1 or 2 questions to use on the next exam. And by the way, our next exam is Wednesday. And the review will likely be Monday. And yes, we have a song. Today's song is a very fun song. It's called the sound of glucose. Aldehyde sugars are always aldoses, and if there's a ketone, we call them ketoses. Some will form structures in circular rings. Saccharides do some incredible things. Onto a glucose, we add a P to it. ATP energy ought to renew it. Quick rearranging creates F6P without requiring input energy. At a high rate, add a phosphate with PFK. F16BP is made up this way, so we can run and play. Aldolase breaks it, and then it releases DHAP and a few G3 pieces. These both turn into 1,3-BPG, adding electrons onto NAD. Phosphate plus ADP makes ATP, while giving cells what they need, energy, making triphosphates a situation of substrate-level phosphorylation. 3-BPG, 2-BPG, lose a water. PEP gets a high energy state just to make pyruvate. So all the glucose gets broken and bent. If there's no oxygen, cells must ferment. Pyruvate, lactate, our cells hit the wall. Some lucky yeast get to make ethanol. This is the end of your glucose's song, unless you goof up and get it all wrong. Break it, don't make it to yield ATP. You'll save your cells from futility. Okay, guys.